Okay, welcome back, folks. We spent a lot of time in class today. You spent a lot of time in class today looking at how to figure out how to do three-point test crosses, and that's very well time spent. But that means that we didn't get through all of our slides, all of our content today. So I'm going to wrap that up here. My camera's a little bit off kilter, so sorry if I'm not kind of right in front of it. I'm off to one side, so just bear with that. Okay, we can also use what we learned about recombination and recombination frequencies to make predictions about gamete frequencies. So the relationship between recombination frequency and map distance can be used to predict the frequencies of recombinant gametes based on the map distances you calculate from the recombination frequency. So we have a situation where we've got an organism which is big A, big B, linked and not completely linked and completely linked, and that's organisms also little a, little b on the homolog. And let's just say that those genes A and B are 10 map units apart, 10 centimeters, 10 centimorgans, or 10% apart. So when that happens, remember, we're going to get two parental gametes and two non-parental gametes produced. And so the recombinant gametes are going to be expected to be produced 10% of the time. That's just what our recombination frequency is. We work backwards from the 10 centimorgans or 10 map units to a recombination frequency of 10%. So from that, we can ask ourselves, okay, what frequency of gametes should I expect to see? So the recombinant gametes, that would be big A, little b, and little a, big B, they should be observed equally frequently, and they should be observed half of 10% of the time for each outcome class in terms of the gamete. So an outcome class here would be big A, little b, or little a, big B. And so half of 10% would mean that down here you can see that this recombinant gamete with this recombinant chromosome would occur half of 10% of the time, which is 5% of the time, 0.05, and the other recombinant gamete, little a, big B, would be produced half of 10% of the time, it's 0.05 or 5%, and so then the parental gametes would be produced 45% of the time each. In combination, that would sum up to 90% of the time. So we can use our map distances that we get from recombination frequencies to figure out what the proportions of parental and recombinant gametes would be, which is a neat little trick. Okay, what about if we have three genes and we want to think about the gamete frequencies or the proportions of gametes, the relative abundances? Well, remember you're always going to have 8 outcome classes here. So we're going to have, like we see here, two parentals there. We've got these two single recombinants, these two single recombinants, so that's 2, 4, 6. Then we've got this double recombinant, so 8 outcome classes. How do we work out what the gamete frequency would be? Well, if we let's just say that there is there's a 10% chance of recombination between A and B, and a 20% chance of recombination between B and C. So what is the probability of recombination between A and B? 10%. Therefore, what's the probability of no recombination? 0.9. And what's the probability of no recombination between B and C? 1 minus 0.2 would be 0.8. So if we multiply 0.9 by 0.8, that gives us 0.72. We divide that by the two types of parental gametes, and you can see that each of the parental gametes would be produced at a frequency of 0.36. You do the same thing for the recombination frequency between A and B and C, and you get the single recombination frequencies. So you can do that with all of the singles, and then you can do it for the double recombinants too, and you add them all up and equal 1. So that's how you use the product rule to figure out what the recombinant gamete frequencies would be. Just bear in mind, your book is going to talk to you a little bit about the something called interference, and that relates to the frequency of the double recombinants. And there are phenomena which produce this this observation called interference, where your actual double recombinance, your actually observed double recombinance, will be much lower than it's predicted. So if we predicted 0.01% and 0.01% for these double recombinants as 2%, there's a good chance you're going to see much, much lower than that in the actual observed double recombinance. So you can calculate what the interference is between in the double recombinants and whatever use that might be to people. So that's interference. You'll read about that, I'm sure. Okay, so don't forget that we get crossover leading to recombination in early prophase 1, and that process completes by the time you got to metaphase. So what's happening is that in prophase 1, you establish these points of crossing over, what are called recombination nodules, along the parts of the chromosome which are synapsed, and those are called the synaptonemal complexes. So remember that the closer the genes together, the less likely you can form one of those recombination nodules. The further apart, the more likely you are. So we have this phenomenon that the recombination frequency is proportional to the distance between the genes. Open it up, greater recombination frequency. Close it down, less recombination frequency. So don't forget that. So the actual event of crossing over occurs after S phase when we go into meiosis. Excuse me, it's hard. I had a sore throat after general biology all afternoon. And so we get the crossing over event at the point where the homologs have got replicated sister chromatids. Remember, as you go into S phase, you take your single chromosomes, you do S phase, and that produces a chromosome, a homolog, with a pair of sister chromatids. So that means when you've got a pair of homologs, is what you're actually doing is you're ending up with four strands of DNA. And the recombination occurs that between some or all or none of those 4 strands. Okay, so that's what we're going to look at next. We're going to look at how you get single and double crossovers and that kind of thing. So that's what we're going to go for now. Okay, so when we look at these crossovers, when we have a single crossover, remember you're going to get 2 recombinant and 2 parental gametes that are formed. So that's 50% and 50%. Now, bear in mind, there are different ways that you can do recombination between the 4 strands. So these are the 4 ways that are going on here. There's nothing fancy here. All we're doing is we're picking different sisters to do recombination from. So let's just look at this first class here, and then you can work through the other 3 classes on your own. Once you understand it with one, the others you can figure out. So what we've got going on here is we've got recombination occurring between this Botta system of what is meant to be the paternal homolog at the top here, and we've got recombination between this top sister of this what is meant to be maternal homolog. And so what we're going to do there is when we do meiosis, we're going to end up with a gamete which has got this upper paternal chromosome, and that's going to be a parental because nothing's happened to it. We would get a gamete with this bottom parental, which is the maternal, and that's this pair here. So half of the total 4 that we're going to get are going to have a maternal or a paternal. So just bear in mind that these would be separated during meiosis 2. So they'd be separated during meiosis 1, then we do the sisters in meiosis 2. But if we have a crossover between one of the maternal and one of the paternal sisters, then what we're doing is we're going to switch out the alleles. We're switching this little b for this big B here, basically, when we do this, and that gives us our pair of recombinants. So you can work through how this happens with the different sisters being used, and it's the same outcome. Doesn't matter whether you use the top sister from the paternal and the top sister from the maternal, or the top sister from the paternal and the bottom sister from the maternal. You get this frequency of, for every 1 recombinant gamete, you get a parental gamete, and that is just, that's just the ratio you're going to get when you do this. You're going to get 4 outcomes, 2 paternals and 2 recombinants, and obviously the frequency by which you get the recombinants and the paternals in the gametes. When I say paternals and maternals and parentals, I mean the sisters, which are ultimately chromosomes from a homolog. Obviously, that frequency is determined by the recombination frequency between the 2 genes that we're looking at. So you're not going to get 50 and 50 all the time. You're going to get some value between 0 and 50 in terms of your recombinants, and then the value for your paternals is going to be much, much higher than that. So, and that's a reflection of that notion that the further the 2 genes apart are, the higher the recombination frequency is. Okay, so we can now look at our double crossovers in a little bit more detail. Now, when we do double crossovers, there are three ways we can do double crossovers, and so this gets a little bit fussy. We can do two-strand double crossovers, and that will only produce parental gametes, only parentals from the two-strand doubles. We can do three-strand double crossovers, and you'll see that that will produce 50% parental and 50% recombinant. And then if you do four-strand double crossovers, everything will be recombinant gametes. So we can look at each of those really quickly. Now, as we go through this, I just want you to develop familiarity with this. I'm going to write exam questions on this. I want you to have a familiarity with the actual phenomena of crossing over, which is why we're doing this. So you might have to think about this a little bit on an exam question, but the exam question is not going to directly examine this concept. Okay, so we've got a two-strand double crossover here. So what we're doing here is we're going to take the two strands. Here are our two strands. This lower strand from the paternal, this upper strand from the maternal. And if you actually look at what's happening here, there is actually an exchange of genetic material. But what you're going to do is switch out this middle blue piece with this middle red piece. Now, when we do the 3-point crossover, if you put a gene in here between A and B, so if we had gene A, and then in the middle here we had gene Q, for example, and then we had gene B, and then down on the maternal, if we had little a, little q, and little b. Now, if we did this, and we did a double crossover, that's when we switch out the middle gene, but there's no gene here that we're looking at. So although you get an exchange of genetic material, we're not switching genes out. So what you end up with is all of your gametes being parental, even though you have had an exchange of genetic material. You've not switched any genes. So this will obviously depend on the locations of the genes, but in the situation here, then there's no gene being switched out. And so you can see that if you separated these two and you did meiosis 2, you'd have a parental, which would be big A, big B, which is what you got over here. You'd have a parental big A, big B, which is this one down here. You'd have a maternal, which would be little a, little b, which is this one. They're the same, right? You don't get any recombinants. So you've got 4 outcomes here. If you do meiosis 2 with this arrangement here, you end up with 4 gamete types, but none of them are recombinant. No recombinants out of the 4 from that kind of double crossover. Let's look at 3-strand double crossovers. So what we're doing here is we're doing a crossover between homologous regions on this upper maternal chromosome, and we're doing one crossover with this lower paternal chromosome, and we're doing one crossover with the upper paternal chromosome. So this is still a double crossover because there's one crossover here and one crossover here. It's just we're using 3 strands. Remember, you've got 4 strands in a pair of sister, in a pair of homologous chromosomes. You've got 4 sister chromosomes. So, 4 sister chromatids. So when you figure out what's getting crossed out here, is what you see is that if you do the 3-strand double crossover with this arrangement at the top, you're going to find that you get 2 recombinants out of a total of 4 gametes. So what are our recombinants? Well, what we've got is we've got this big A and this little b, which has resulted from switching down here. And we've got this little a and this big B, which is resulted from switching up here. And so this one here, this big A, little b chromosome, that will end up being our gamete that's recombinant, and this little a, big B would be our recombinant. How do I know they're recombinants? Well, because all of the parents are big A, big B, or little a, big B. So our parental chromosomes are big A, big B, and little a, little b. So recombinant frequency there is 2 out of 4. You can also do it the other way around, where we use the 2 sisters from this maternal chromosome, and then one sister in the paternal chromosome, and you get the same phenomena. But if you look at this carefully, all you're doing is switching the colors around. It's just a reciprocal arrangement, basically. It's just the opposite of what you did at the top. And so half of all the gametes there are recombinant also. So that's our 3-strand double crossover. We can also do it this way. And again, now when we do it this way, it's still a 3-strand double crossover, but now we're using all 4 of the sisters. Okay, So we can do a recombinant event here between this lower paternal and this upper maternal, and then we can do a recombination event between this upper paternal and this lower maternal. And when we do that, is what we end up producing is all of our gametes would be recombinant. So in that situation, we can have all recombinant gametes. How do we get that? Well, none of these gametes are the paternal genotype. None of them are big A, big B, and none of them are little a, little b. Okay, so that's our four-strand business. When you look at all of these single and double crossovers and the way you can do it, is what we find is that we can get 8 recombinants out of a total of 16 possible outcomes. And if you back up a couple of slides, is what you'll see is, okay, this is 0 out of 4. Okay, now it's 2 out of 8. And now it's 4 out of 12. And now it's 8 out of 16, which gives us this 50% recombination frequency that we've touched on before. And so you can see that summarized here. The 2-strand double crossovers, they're going to give you 0 of 4, 3-strand 2 of 4, 4-strand double crossovers 4 of 4 recombinants. Now, you only get this 50% value being reached when you got the genes really, really far apart. So just keep in mind that relationship. Okay, last topic to look at here, and again, don't spend too much time on this. This is more risky in terms of content that you may find in your exam. We're going to look at how you map genes relative to their centromeres, because it's what we saw is that you can map A, B, and C using a 3-point test cross, and you can map B to the middle, but you don't know whether A is on one side and C is on the other, or it's flipped. Remember, ABC is the same as CBA when you're doing a 3-point test cross. All you can do is put a gene in the middle. So you can actually map genes given their proximity to the centromere of a chromosome. And you can do that using certain techniques in haploid organisms, and it involves what's called tetrad analysis. So we'll start off looking at unordered tetrad analysis, and you'll understand what an unordered tetrad is in a few moments. This is commonly done in certain groups of fungus. So ascomycetes. They're a big group of fungus. They're if you've ever seen a fungus which is growing kind of like it's got a cup on the top, that's an ascomycete. They're also called the cup fungi. Don't get me started on fungus. One of my book chapters is on fungus, and so I spend a good amount of my year reading about and learning about fungal phylogeny. And believe me, that's not so much fun. But so let's not get too into fungus. Now, some of the organisms in the ascomycetes, they actually have a really interesting life cycle. They have a haploid stage and a diploid stage in their life cycle, and they can exist perfectly happily in both of those kind of life cycle stages in the environments. That feature is exploited to do these analyses. So let's look to see what happens in a yeast. And this is literally like this is beer making yeast or bread making yeast. It's Saccharomyces cerevisiae. So if you open a loaf of bread and it's got a kind of a rustic smell to it, you're probably getting some of the alcohol that's not burnt off, but also probably some of the secondary metabolites produced by the yeast. So you're familiar with the yeasty smell. Okay. Yeasts, Saccharomyces cerevisiae, can live in the environment in 2 forms, and they're mating types. And the mating types are A and alpha. So A cannot mate with A, alpha cannot mate with alpha, but A and alpha can mate. I know, it's weird thinking about yeast reproduction. So in the environment, the A yeasts are unicellular, and they will quite happily just undergo mitosis as a haploid. They just got a haploid number, they're N, and they will quite happily live, and they go through this process of growth, and they will grow, and they'll produce this little bud, and the bud will grow, and you get this structure here is actually called a shmoo. So if you don't know what a shmoo is, if you didn't grow up in a certain time in the 80s, you won't know what a shmoo is, but Google shmoo, SCHMOO, and you'll see what a shmoo is. So this structure is called a shmoo, after an 80s cartoon character, basically. Alpha is the same thing. It will cycle, it will go through mitosis, it will go through G1, S, G2, mitosis, and it will just divide in the environment or in a flask in the lab as a haploid. And so these are the two mating types. Now, if you mix the two mating types together, they will fuse. So the A and the alpha fuse together, and you get a diploid. Now, the diploids, under a lot of circumstances, they will just happily go through mitosis. So they fuse, and then they will just go through mitosis. They will just go through mitosis as a diploid. So they will just do their regular G1S, G2M, very well studied, very well understood cell cycle. So they will do this quite happily. Under certain conditions, the diploid will undergo meiosis, and you, like in the meiosis that you're familiar with, you'll undergo S phase, and then you'll go through a G2, and then you'll do a meiotic division, and then you'll do another second meiosis without an intervening S phase, and that will give you the 4 cells that are haploid that you're familiar with. And so if this diploid, this A alpha, undergoes meiosis, you end up with this structure which is called an ascus, and in the ascus are 4 spores, 2 alpha and 2 A. And so that is called a tetrad because there's 4 of them. And when you look at these asci, you can't determine the order of the spores. You don't know whether they're A or they're alpha, and that they're not arranged in a particular order. And so they're said to be unordered tetrads. But you can still germinate these. You can take the spores and you can put them on a plate and you can germinate them, and then you can determine their genotype. And you can look at the phenotypic characteristics too. And so that whole process is called tetrad analysis. By you mix an alpha and an A culture together, and you get the diploids. And the diploids, you can induce them to undergo meiosis. You stress them, you starve them, that kind of thing. They go through meiosis, and they produce an unordered ascus or an unordered tetrad. And then you can look at what these spores are, whether they're A or they're alpha. So that's the background to this, really. You can look to see what is happening during meiosis in the production of these tetrads. So when you have a diploid yeast cell, and let's say it's a dihybrid yeast cell, and it's got the genotype A plus A, B plus B. So here would be the 2 chromosomes in our dihybrid. We're on separate chromosomes now, so kind of put genetic linkage to one side. We'll come back to it. If we've got 2 genes on 2 separate chromosomes and we're in a diploid state, we've got A plus and A, and then B plus and B. If these undergo, these 2 chromosomes undergo meiosis, we do meiosis 1 and meiosis 2. Is what we end up with are you can figure out what the gametes would be, and then when you get the tetrad, you would end up with a known or a predicted outcome in terms of the genotypes of the spores. And when the spores, one of my dogs is slurping, sorry. When you look at the genotypes of the spores, is what you see in the situation we have going on here, which is just independent assortment of chromosomes A and B. We're going to end up with 50% recombinant gametes, and then, we're going to end up with 4 basically parental gametes. They're parental because we've got 2 ABs, which is what we started up over here, and we've got 2 A plus B plus, which is what we had over here. So that's called a ditype, and independent assortment here is going to lead to this parental ditype here. Sometimes got a diplotype as well. Okay, excuse me, I've got to have some water because my throat is burning. Okay, so that assumes no crossover up here. Okay, and that gives you your parental ditypes. Alternatively, if you do independent assortment differently than we did at the top here, then we can end up with this configuration of the gametes of the spores. Basically, the spores are effectively the gametes. They would germinate. They'd be haploid. They don't fertilize, but they would go into the haploid life cycle. So those are your 2 alternatives without any crossing over. Now, what about if we get crossing over? Now, if we get crossing over between homologous chromosomes, then we're going to start to get some diversity. And so if you see what the chromosomes, if you track the crossover that you've got going on here with chromosome A, that would be our first option, a crossover on chromosome A. You can see what the gamete products would be here, and you can then see what they would end up being in the tetrad. And so this A plus, B plus here is this top genotype, so that's a spore. Then this second one here would be A, B plus, that's that second spore there. And then this third outcome here would be A plus B, and that's that little spore there. Then the bottom one here is A, B, and little a and little b. And so this bottom spore is parental. This top spore is parental, but because of the recombination event over here between the sisters on this first homologous chromosome A, we end up with these two recombinants. So here, the tetratype is referred to as being two parentals and two recombinants. So you can see in the tetrad whether there's been recombination or not. And if you compare that going back one slide, you can clearly see what you'd expect given this independent assortment versus this independent assortment versus this crossover in chromosome A. And so if you follow that through, you can see you'd get exactly the same thing, but basically reversed if you did a single crossover with chromosome B. And so this top genotype of this spore up here in the tetrad would be this top parental chromosomes, and then down here we have this parental AB spore coming from these two gametes, coming from these sisters over here in the parents, and then our two recombinants will just be the opposite of what we saw up here. So you can look at this is pretty. It's actually kind of easy to do. You can do this in undergraduate teaching labs. You just take the two alpha strains. You take the alpha and the A strain, and you mate them, and then you look to see. Okay, I've got two marker genes which you can determine physiologically or you can determine morphologically, however you do it, and you can measure quite easily using unordered tetrad analysis what the frequency of recombination is, because obviously the frequency with which you get these two tetrads here is directly proportional to the frequency of these recombination events. And if recombination is 0, then you're going to always get these two ditypes. If you ever see these two tetratypes, you know there's a recombination going on, and the frequency with which you see these arrangements tells you what the recombination frequency is between the two genes. So that's kind of neat. Now, what about if we have linked genes? So now we make it a little bit more complicated. Now remember that if we've got linked genes, then the parental alleles, they will always segregate together preferentially. And if they're so close together, you're never going to separate them, and all you will get will parental ditypes. And that's what's happening up here. Here, we've got no crossover at all. And so these, if we separated these in meiosis 2, then our tetrads would be basically all the parentals. We would have little a, little b, little a, little b in the products, and here's that one, little a, little b here, little a, little b in the gamete, and there's that product. And so this would be no crossover between linked genes. Now, if we look at a single crossover event here, again, bear in mind is what we're doing is we're switching out this B plus here with this B on this lower paternal chromosome. And then this would undergo meiosis 2, and we would end up with a paternal chromosome, which is little a, little b, and that's the spore. We would end up with this recombinant chromosome, A, B plus, and that's that spore there. This chromosome here would separate from this one down here, so this recombinant would separate, it would go into a gamete, and it would be A plus, B, and there's that one there. And then lastly, our parental would be A plus, B plus, and there it is there. So once again, you can see that the frequency of the crossover events here will be reflected by the frequency of this tetratype, two parentals and two recombinants. The more of these arrangements you see, then if you figure out you've got a linked gene and you see these 22% of the time, that tells you something about the recombination frequency. Now, if you've got a double crossover, now here we've got a two-strand double crossover, and you can think about other types of crossovers as well that we've talked about before in the last little segment. If we've got a double crossover with two-strand, now it's the same situation before when there's no gene in the middle here to be switched out. What we end up with is basically the parental ditype. So yeah, there are double crossovers here, And so that indicates that there's recombination happening, but we can never detect it. So effectively, this double crossover down here is being recorded as the same event as though there was no crossover, because the tetrad you get up here is the same as the tetrad you get down here. So you don't learn anything about the recombination frequency here. There's recombination happening, but it's not affecting the genes you're looking at. And that may mean that you want to start looking at another gene that's in here that would actually reveal the recombination frequency. If this is so far apart and you get these two events, you're never going to detect those two events, even though there's a recombination event. Remember, you can only detect a recombination event if you switch genes around, and then you can detect that in the phenotype and make a conclusion about the genotype. So I will let you walk through how you can do the double crossover, and we've got two double crossover mechanisms here using the three-strand models, and you can figure out what the gametes would be and make them correspond to the spores. So once you've worked through those two and you've figured out what the spore phenotypes and genotypes, phenotypes and the spore genotypes can be, you can basically end up working out what the recombination frequency is. So the formula that's used to determine the recombination frequency in tetrad analysis is similar to that with what we've done before. So there's relationships here. The number of recombinants is divided by the total offspring, and because only half of the spores in the tetrads are a recombinant, we use this formula where it's half TT plus NPD divided by the total tetrads, and that gives you the recombination frequency. Okay, I'm not going to have you do this. I'm not going to have you counting spores, so don't worry about it. The worst thing you're going to have to do on an exam is look at, for example, discriminate between, for example, this tetrad and a tetrad like this one, where you can clearly see that there's been recombination. So the worst thing that I could ask you to do is to tell me, hey, look, no recombination, but in this tetrad, you're seeing recombination, and to explain why based on the recombination event. That's the worst thing you're going to have to do. You're never going to have to be counting spores up. It takes forever. It's annoying. Okay, ordered ascus analysis. Other types of ascomycetes produce an ordered ascus. So how do you get an ordered ascus? So you have these two mating types as you do before. You saw that with the yeasts. You have these two mating types, A and A+. You saw A and alpha before. There are just these two mating types, and here we're indicating the genotype is A and A+. They're just two haploid cells. They form to what's called a myocyte. And so if you're taking an N cell and an N cell, there's your diploid. And that diploid undergoes meiosis 1. So what would happen is you would segregate A+ from A, and there's what you get there. That's your first meiosis. In your second meiosis, this would actually be A+, and then you would separate the A+'s out, and that would be your 4. Now these asci actually form these. They're long, very long, thin oval structures. They're not spherical like the unordered asci that we just saw. So when this spore here undergoes the second meiotic division, when it divides, it puts 2 of the same genotype right next to something, and then something a little unusual happens. In these types of yeasts, each of these 4 spores undergoes a mitosis, and so you end up with 8 spores in the ascus. So if you take this spore here and it divides into 2, you get these 2, and then this A+ spore here and it divides, it's these 2 here. This A becomes this pair, and then this last A here becomes this bottom 2. So what you see is that the order in this ascus reflects what happens before mitosis and what then happens between meiosis 1 and meiosis 2. And because these divisions are right, that they divide and you get 2 cells right next to one another, that's what gives you the ordered phenomena. You can take these asci, you can break them apart. You can take the spores, you're doing this under a microscope, you take the spores, you can genotype them or you can germinate them, and you can look at the phenotype and make a conclusion about the genotype, and they're haploid. Remember, you've done a meiosis 1 and a meiosis 2, and then you're just doing a mitosis on a haploid So everything you get is haploid. So basically, whatever phenotype you see, it's due to just one allele of the gene. You don't have to worry about being a diploid with the second gene interfering or masking the effect of another allele. So that makes the analysis a little bit easier. So what do we do with these then? You can look at the order of the ascus, and you can use that information in the order of the spores in the ascus to actually calculate the distance of a gene from the centromere. So the value of that is that if we need to know the order of A, B, C, and we know that it's A, B, C, or CBA, if we can determine out whether A or C is closer to the centromere, we can then actually say, well, it must be ABC rather than CBA. Okay. So the first analysis you can do is you can do what's called you can look at the first division segregation pattern. Okay, this isn't actually that useful, so don't linger on it too much. So if we've got our diploid organism here, we've got this pair of homologous chromosomes. It's heterozygous in respect to the A gene, so we've got A plus and A. Now, if we do meiosis 1, we separate the homologous chromosomes. So there's the A plus chromosome, there's the A chromosome, and then if we do meiosis 2, we can determine that these two spores here must have obtained this A plus from a certain chromosome over here in the parent. That doesn't actually help too much. We actually have to look at what happens in meiosis 2. So here, the pattern you're seeing over here is because of the segregation of alleles during meiosis 1. But if there's no recombination, you don't really learn anything. So what is more informative is that you can learn something about the distance to the centromere of an allele using what's called second division segregation patterns. So if we have a heterozygous myocyte and we have a crossover that occurs between the gene A and the centromere, when the sisters separate over here in meiosis 2, you can determine basically based on the frequency of these crossover events over here and the pattern of what you get in the ascus, the spores in the ascus, you can determine the distance from the gene to the centromere. Okay, so you do this called what's using a second division segregation pattern. So we do a crossover between these two sisters here, so it's a single chromatid crossover, it's a single crossover event. We end up with our homologous, our homolog here with its paternal parent, sister, and then we've got its recombinant arm here, and then in this chromosome, this homolog at the bottom, we have the situation where we've got the recombinant chromosome, and then the other sister is the parental, and it's the maternal given the color coding that we're looking at. So when we do meiosis 2, you can see how the different sisters separate to give us this particular pattern in the ascus. Now, depending on how you arrange the chromosomes in meiosis 1, you can get these different patterns of spores in the ascus, in the asci rather. So the way that we're looking here is we've got this parental chromosome kind of at the top, and that produces a spore which then undergoes mitosis to give us this 2 right next to one another. And then we've got this A plus recombinant chromosome. It produces one of these spores, and that goes mitosis and produces these 2 red ones. And then this little a recombinant chromosome divides to produce this spore. It segregates and it becomes this spore, and then that undergoes mitosis to produce these 2. And the same thing happens with the A plus. You get this genotype in this spore, and then you get mitosis to produce the duplicate ones. So if you think about it, if you sorted this sister not into one of the top gametes, the spores, but into this one here, when it undergoes mitosis, you get this pair. And if we sorted this A plus into one of these top spores, so just flip these 2 around, just flip the recombinant with the parent around, you would get this pattern. Okay, and then you can figure out how you get these different patterns based on how you separate the sisters in meiosis II. So the way you separate the sisters in meiosis II gives you different patterns in the ascus, and all you're doing is looking for phenotypic markers. The classic one is that you can either get pale spores or brown spores. So under a microscope, you don't even need to get the spores and germinate them. You just look down your microscope, and you're just looking for different spores in different patterns. And so you just have a little chart, and you say to yourself, okay, is it this arrangement? And you make a mark. And is it this arrangement? You make a mark. The problem is you can't tell these two apart, right? Because if you flip this first one upside down, it's the same as this one. So actually, it was all you're ever scoring of these ones versus these ones, because you can't really tell these two apart. You literally look down your microscope and you just look at the spore pattern and you're like, oh, that's a type 1, oh, that's a type 3, and that's a type 4, and you just make a little tally chart. And that gives you a pattern of second division segregation. How do you use that data? We go back to our recombination frequency, and that's equivalent to the maps. And is what we do is we look at the half of the number of second division segregation asci versus the total number of asci. So you're saying, okay, how many patterns do I see like this as a function of all the asci that I see? And then multiply that by 100, and it gives you your recombination frequency. Okay, so let's kind of look at an example. Let's just look at this. Let's look at this parental genotype here. It's big C, C plus, G plus, and then the recessives down here, C and G. And the trait here is color. This is probably the pale and the brown spores. And so you look for your first division segregation patterns. So you look to see how many of these do I get. When you got that, there's no recombination. You don't have any recombinations between the centromere and the A plus. So there's no recombination going on here. So those are all basically your parentals. When we talked about 3-point crosses, those are your parental gametes. Basically, not gametes, obviously. Each of those is a gamete, but you see what I mean. And so then, let's say your first division segregations are 73. You count them up, and you've done your cross, and you've done your mating types, and your first division segregation patterns are 73. And then you count up and you say, I've got 36, and you're just looking at color. So you're just going through looking, are they brown, are they pale? And that gives you say, okay, when I counted these up, I find 36 of these variants, 73 of those variants. That means your combined total is 109. You take your second division segregation patterns divided by 2, so that will give you 18. 18 divided by 109. And you convert that to a percentage, and that gives you 16.5. That's the recombination frequency. You just convert, it would be 0.165, and that would be 16.5 centimorgans. So what that tells you is that the distance between this gene and the centromere is 16.5 centimorgans. And if you do the same for the other allele, you can calculate that would be, in this case, 30.7 centimorgans. So that's how you would do an ordered tetrad analysis to determine the distance between a centromere and one allele, and a centromere and another allele. Okay, so you can also probably figure out that you can calculate the distance between C and G also using this approach. But you'd have to look at both characteristics. But you could do that if you wanted to. Let's not. Okay, the last thing to talk about. We've been talking about meiosis. 99.99% of what we worry about is the products of meiosis. But it was discovered that with a very, very low frequency, this is very low frequency, you can actually get recombination during mitosis. And that is just because the homologs come close enough together when they're being moved around sometimes that you can get a recombination event going on. This was discovered in Drosophila. It's actually clinically important. It happens with a high enough frequency that it can be clinically important. When I say clinically important, what I mean is that it can be used as a way to create a second mutant allele in a cancer cell. So if you think about the frequency of mutations, and if we say the mutation frequency is 10 to the 6 or 10 to the minus 6, that's really low. So to get a second mutation in a tumor suppressor gene, to get 2 recessive alleles, remember how we said that means the frequency is going to be 10 to the minus 12, and there just aren't enough cell divisions in your lifetime, in your body, to actually have that many mutagenic events. And so it turns out that the way that you can lose a second allele is through this phenomenon of mitotic recombination. So this happens very rarely, but it happens much more frequently than does mutation, that you can actually take a mutant allele in one chromosome and switch it out with the other allele in the other chromosome. And the way that, if you do that in certain ways, you can actually lose that second allele. And so it becomes clinically kind of important. So I do a lot of that stuff in my cancer biology course, looking at mitotic recombination as a mechanism for driving tumorigenesis. Okay, so this was figured out by a guy called Stern, and he was doing crosses with dihybrid female flies. And when he was doing these crosses, he was doing these crosses with genes called singed and yellow. And if you do this cross of a singed fly that is gray-bodied, and so if you have this effectively dihybrid where you've got singed wild type and singed the mutant, they're normal. They've got normal hairs. The singe this is actually a really cool little phenotype. Actually, it looks like someone has taken a match to the Drosophila, and on their body they've got these little tiny hairs. So if you've ever burnt your hand or your arm on a candle, you know how your hairs all kind of frizzle up and they get all singed. It looks like that. You get these little flies and they've got these little sad singed hairs. But so this organism here, this fly here, is singed wild type, and then it's got one mutant allele. So its hairs are normal, and then you've got this yellow allele and the yellow wild type allele, and so that fly should have normal hairs, and it should have a grey body. And so when Stern was actually crossing these flies, he found that what he was expecting is that they should all be fine. They shouldn't have any weird phenotypes. And so what he found was that with a certain frequency, you get a single yellow spot. So you'll get a region on the body of the fly which is not grey, it's yellow. You can also get a frequency. You can also, with some frequency, get a region of the body which has got singed bristles. And then at an even lower frequency, these are really low frequencies, very low frequency, he found that you could get a spot of yellow right next to a spot of singed hairs. And these were called twin spots. So this is a twin spot. This graphic is terrible, actually. That's meant that bold spot is meant to be yellow. You can see it a little bit better here, but I don't know what this artist was thinking. They should have a much darker background or make the patch here much yellower. But these are meant to be yellow patches, and then that's a singed patch here. So here is what's going on here. Let's just figure out what's going on with the twin spots. So when you have this chromosome up here, which is described over here on the left, the genotype is over on the left, is what's happening is that with a very, very low frequency, you get a recombination event between the singed gene and the centromere, which produces these recombinant chromosomes. And when you get this, a certain frequency of the offspring end up being singed mutants and yellow mutants. And that's happening because of mitotic recombination. This is in the body of the fly. We're not talking about gamete production here. We're talking about an event that must be happening in mitosis. And so what you end up with here is you end up with a clone of cells which are yellow and a clone of cells which are singed. So the yellow cells were derived from a cell which had two yellow alleles, and then the singed patch here is being derived from something that's got the two singed alleles. And so, because we're not dealing with meiosis here, the only way that could happen is if you had homologs coming close enough together during mitosis and actually crossing over during mitosis. And so this happens with a very, very low frequency, but like I say, clinically important because it happens with a frequency greater than single mutations. So if you need to mutate a second tumor suppressor allele, you can't do it just relying on mutations. It's extremely unlikely, but you can use mitotic recombination, and that happens with enough of a frequency that this phenomena can drive tumorigenesis. Again, not anything super important for us, just a neat little phenomena that you should know about. Don't worry too much about the crosses. Don't worry too much about the chromosomes here. Just be aware that there's this phenomena where you can get recombination during mitosis, not just restricted to meiosis. We're going to focus exclusively on meiotic recombination for the rest of the course, and everything we've done before this is meiotic recombination. Okay, that's it. My throat is done. I will work on getting the materials up as soon as I can so you can get ready over the weekend. Have a great weekend. I will see you on Monday. Keep working hard.